Start recording, share my screen. So today is August 10th, and we're going to be working on Chapter 8, Microbial Genetics. Please note you do have a quiz this Wednesday, and it will be quiz number 4, covering chapters 5 and 6 and lab 7. Any questions about any of that? We do have our last lab tonight, so somebody please let me know when it's, what time, 6 o'clock? So I can wrap down the lecture, and then we'll reassemble for 6:30. I'll start with a lecture, but then we'll move on to the lab, and that lab will be lab 9, the identification of gram-negative organisms. Please note your infectious disease paper is due at 11:59 p.m. this Saturday. And I haven't replied to a few emails. There's a few students who have asked me about topics, and I'll try and get to those in the lab. All right, any questions about it, what we're doing? I guess I can leave this up for the lab. Take out my thumb drive and put on my headphone. I haven't heard anything. Is everybody hearing me okay?
I can hear you.
Great. Let me check my volume.
Hey, Dr. Volpe, are we supposed to have received some results yet for our unknowns projects?
Yeah, you should have received the first 3 tests. The gram stain, the gelatin hydrolysis, and the PR lactose test. I emailed those results. I don't remember when.
Right. Yeah, I got those. No, I'm talking about for in the unknown notes, like when we submit for tests, because I submitted one a week ago. I haven't seen anything.
Did you request the MR test? Hello? Did you request the MR test?
Is that the methyl red test?
Yeah, the methyl red test.
Yes, I did.
Okay, that one incubates for 5 days, so you're not going to hear anything for 5 days. Okay? I knew there was, I think, 2 people who requested that, and those I have not sent out. You might be now, I just sent out results last night, but you might be getting results today. I don't know. I haven't looked when I'll be sending out the results to that. But you have to look up how long the tests take to incubate, and you're not going to get results before the incubation period. All right, we had talked about this slide last time, DNA replication. I think we need to start talking about, oh, actually we have talked about this slide too. This is DNA replication. All right, any questions about DNA replication? If not, let's move on to DNA transcription, where the DNA is read and transcribed into RNA. DNA is transcribed to make RNA, and there are 3 types of RNA. Each different RNA will be made at a different gene. If the gene codes for a protein, the gene will be transcribed as messenger RNA. If the gene codes for tRNA, the RNA will be transcribed as tRNA. If the RNA codes for rRNA, the RNA will be transcribed as rRNA. It's as simple as that. Any questions about any of that? Most of the genes code for a protein. So most of the genes will be transcribed as messenger RNA. However, if we look to see what the majority of the RNA is in a cell, the majority of the RNA is actually ribosomal RNA or rRNA. That's just something you have to sort of remember. Only the genes are transcribed. When we're talking about DNA replication, all of the DNA is replicated. When we're talking about transcription, only the genes are transcribed. All of the DNA that does not encode something, meaning it's not a gene, will not be transcribed. And in eukaryotes, that is the majority of the DNA. Any question about any of that? So in eukaryotes, the majority of the DNA will not be transcribed because it is not a gene. In prokaryotes, the majority of the DNA is genetic material, meaning it is a gene. How transcription begins is RNA polymerase binds to the promoter region of DNA. And then the RNA polymerase moves down the DNA in the 5' to 3' direction, transcribing the DNA, making RNA. So RNA is made in the 5' to 3' direction. Transcription stops when the RNA polymerase reaches the terminator. The terminator is a region at the end of a gene that codes for the RNA polymerase to fall off. Any question about any of that? All right. That's a lot in one slide. Here we're looking at an actual picture, I assume of an electron microscope, showing you RNA transcription being made. And the white dot is actually the RNA polymerase, which transcribes the DNA to make the RNA. So remember, transcription is coding copying the DNA and making RNA, a messenger RNA, if that is going to be made into protein. Let's see what I want to talk about here. RNA transcription begins when the RNA polymerase binds to the DNA. The RNA polymerase binds to the promoter region, which is upstream of the gene that's going to be transcribed. And then the RNA polymerase moves down the DNA, transcribing the RNA. How that transcription occurs is the RNA polymerase reads the DNA and then puts on the corresponding RNA nucleotide. So for A, you put on the RNA nucleotide U. For C, you put on the RNA nucleotide G. For T, you put on the RNA nucleotide A, etc. Let's watch a little video on RNA transcription. Oh, Video no longer works. Let's check this one. Well, I have to do a search. I think this is the one I want.
I have ADHD, and it makes it. I'm not an auditory learner whatsoever, which a lot of our lectures.
Proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the message.
Okay, any question about that? I went over a topic that I will get to, but not until the end of the transcription. And that's splicing. All right, let's go back to our lecture. So transcription begins when the RNA polymerase binds to the promoter and then moves down the DNA. As it moves down the DNA, the RNA transcript gets bigger, and the RNA polymerase will do that until it reaches the terminator. The terminator is a region of the DNA after the gene, and it codes for the RNA polymerase to fall off. So when the RNA polymerase falls off the DNA, that releases the messenger RNA or the RNA transcript. And when the two are free from each other, the DNA snaps back into a double helix, by the way. All right, any questions about transcription? I'm going to talk about, well, we already had it in the video. Let me advance. We talk about splicing. There is a complication with RNA transcription in eukaryotes. This does not happen in prokaryotes. For one thing, when the DNA is transcribed, at least in bacteria, there are no introns in bacteria genes. So when the RNA transcript is made, it is the final messenger RNA, and it doesn't need to leave the nucleus because there is no nucleus in a bacteria cell. But in a eukaryotic cell, there are regions of the gene which are called introns. And the introns do not code for protein or for, they do not code for, well, RNA, like a ribosomal RNA or tRNA. They are a non-coding region within the gene. The exons, on the other hand, are a portion of the gene that code for something, meaning the exons code for the amino acids in a gene that's being transcribed as messenger RNA. If the gene is being transcribed as rRNA, the exons code for rRNA. The point is, in a eukaryote, when the gene is transcribed, both the exons and the introns are transcribed. And this is a, what do you call it? Precursor of the RNA transcript. It's not the final messenger RNA or the final RNA. And the introns regions of that RNA have to be spliced out. Splice just means cutting. And so this will be spliced out, and then the exons will be spliced together to make the final eukaryote messenger RNA. The earlier RNA transcript cannot leave the nucleus, meaning the pores in the nuclear membrane will keep out an RNA that have introns. Only the messenger RNAs or the RNAs that are lacking the introns can leave the nuclear membrane. Okay. Anyways, this complication happens only in eukaryotes where the initial RNA has to have the introns spliced out and the exons spliced together to make the mature messenger RNA. Any question about that? All right, let's move back to translation. So after transcription, the messenger RNA will then be translated. Now, if the gene only codes for ribosomal RNA or tRNA, that's all you have. The end product is the tRNA or the rRNA. And so there is no translation for those genes. Any question about that? But for the other genes that code for a protein, the messenger RNA will be translated into protein. The synthesis of proteins occurs at the ribosomes, and they use the information stored in the messenger RNA. Messenger RNA encodes the information in groups of 3 nucleotides that we call codons. Translation doesn't begin at the start of the messenger RNA, though. The ribosome will read the messenger RNA, and it will begin protein translation at the start codon, and that codon is AUG. You should probably know this codon because it's unique, and it's where the ribosome begins protein translation. AUG has 2 functions. It tells the ribosome begin protein translation here on the messenger RNA. And AUG also codes for the amino acid methionine. So all proteins, when they first get started, they will always have the amino acid methionine as the first amino acid. And if the protein doesn't want that amino acid on it, it'll have to later, after the protein is made, it'll have to remove that amino acid. But all proteins, when they're first getting started, will start with the amino acid methionine because AUG codes for methionine as well as starting the process of translation. The ribosome will then read the messenger RNA in groups of 3, calling codons, putting in the appropriate amino acid until the ribosome reaches one of the 3 stop codons. And there are 3 codons that code for stopping protein translation. You don't have to memorize these because there's 3 of them, but one of them is UAA. And when the ribosome reaches UAA, it will stop protein translation. Any question about any of that? So there we're seeing translation happening here. Remember, translation will only happen on the genes that code for a protein. If the gene codes for an RNA, we will never see translation of that transcript. So translation uses the genetic code, and that is the messenger RNA is read in groups of 3 nucleotides that we call a codon. Here you're looking at the codons, and you'll notice that there are only 20 different amino acids, at least in humans, but there are 60 possible codons, greater than 60 actually. I forget what the real number is, but what that means is that there are amino acids like phenylalanine, which are coded by more than 1 codon. For example, UUU and UUC are 2 codons that code for the amino acid phenylalanine. Leucine, that amino acid, is actually coded by 6 different codons. And you can see the first 2 here, and then the next 4 here. There are only a few amino acids which have only 1 codon. Methionine, which we've already talked about, only has 1 amino acid, 1 codon coding for this amino acid. So AUG is the only one. And remember, AUG also codes for start protein translation here on the messenger RNA. The other one, which I'm not going to require you to remember, is the amino acid Tryptophan has only one codon for it. All of the other amino acids have multiple codons coding for the amino acid. So there's more than one codon coding for most of the amino acid. And this is known as degeneracy of the code, where more than one codon can code for an amino acid. Any question about any of that? All right, this is showing you the different steps of protein translation. And let's just look at the video. Going over protein translation.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
All right, any questions about any of that? You do not need to know the names of the different sites in the ribosome. You should know the different molecules that are involved and go over that. So the different molecules involved are the ribosome, and the ribosome comes together. The two subunits come together and then form on the messenger RNA. And then the messenger RNA is the second important molecule, which I guess might be the third if you count this as two molecules for the ribosome. And then the third is the tRNA. It's the tRNA that brings in the correct amino acid. And it's also the tRNA that actually does the reading of the messenger RNA, meaning AUG is read by the anticodon of tRNA, and the UAC actually binds to AUG, meaning the U binds to the A binds to the U, the C binds to the G. This binding is only hydrogen bonding, which, as you know, is very weak. And it is the ribosome which stabilizes the tRNA's binding to the messenger RNA. What happens is the tRNA, not the tRNA, the ribosome assembles on the messenger RNA and then moves down the ribosome, moves down the messenger RNA. I'm having a hard time here for some reason. Sorry about that. The ribosome moves down the messenger RNA in the 5' to 3' direction. And it does not begin protein translation until the ribosome reaches AUG. AUG codes for start protein translation here. And then the first tRNA comes in. And like I said, there's a hole in the ribosome, and it actually binds to the AUG portion of the codon in the messenger RNA. And that's hydrogen bonding that the anticodon hydrogen bonds to the codon. Okay. And then once the messenger RNA or the first amino acid is brought in, which in this case is methionine, because it's AUG, the next tRNA comes and binds in the second spot in the ribosome. And it brings in the correct amino acid for the codon UUA. Okay, any question about that part? The methionine from the first tRNA is then removed from the tRNA and bound to the second amino acid in the second tRNA. This is actually a peptide bond is formed between the methionine and the leucine. What is doing that? It's not discussed in your textbook, but it's actually the rRNA portion of the ribosome is acting as an enzyme to remove the amino acid here and then binding to the amino acid here. And this, as I stated, is the only enzyme that we will be discussing in this class, which is an rRNA enzyme or an RNA enzyme. All the other enzymes we discuss in this class are protein enzyme. Any question about any of that? When the amino acid is then removed from the first tRNA, that allows the tRNA to then be released from the messenger RNA and the ribosome. And it just goes out and will find another amino acid to bind to it, which that would be methionine. And that recycles the tRNA, if you're wondering. The ribosome then moves down the messenger RNA. So instead of this being in the second position, it'll now move to the first position. And that will allow the next tRNA to come in and bind to the messenger RNA. Let me bring this over a little. So the ribosome moves down, and then the tRNA brings in the third amino acid. The growing polypeptide here is then removed from the tRNA. It's only 2 amino acids, so it's not quite a polypeptide, but you get the picture. It's removed from this tRNA and then bound to the amino acid here. That will free this tRNA to be released so that the ribosome can move down 1 codon. And then that will allow the next tRNA to bring in the correct amino acid and release this tRNA will be released. Let me go back. And here's the correct amino acid on the correct tRNA, which binds to this codon here, which isn't even shown yet. And there it is there. The growing polypeptide chain is removed from this tRNA and attached to the amino acid on this tRNA here. And then that will release this tRNA so it can be released. And then the ribosome will move down 1 more codon. And that will continue until the ribosome reaches the stop codon. The stop codon will tell the ribosome stop protein translation. When that happens, the ribosome will dissociate, fall off the messenger RNA, and that will release the polypeptide, which now that it has all of the amino acids attached to it, we will call it the protein. So when the protein is growing, it's not a protein until it gets the last amino acid. But once it gets the last amino acid, we can stop calling it a polypeptide and start calling it a protein. All right, any question about any of that?
How many amino acids do you need for one protein?
Well, most proteins have hundreds or thousands of amino acids. I'm not sure what the smallest protein is, if that's your question. Obviously, like on peptidoglycan, we've got portions of peptides that are put together, but those we call polypeptides, or short peptides. We don't even call it a protein. And I'm not sure what the smallest protein would be, but most of them have hundreds or thousands of amino acids which are attached. Does that answer your question as good as I can?
Yeah, thank you.
Good. All right. Are there any questions about protein translation? Any other questions, I should say? Let's talk about a complication. In both eukaryotes and in prokaryotes, you can have on a messenger RNA, which I guess is shown here, you can have more than one ribosome reading that same messenger RNA, in which case you will have different sizes of the polypeptide, meaning a newly attached ribosome will have a small polypeptide, and then the ones that have been attached for a long period of time will have a larger polypeptide. So in both prokaryotes and eukaryotes, you can have more than one ribosome attached to the messenger RNA. Any question about that part? That's the easy part of the complication, because it happens the same in both prokaryotes and eukaryotes. And what happens is that messenger RNA is in the cytoplasm, and then more than one ribosome can actually bind to it. It's as simple as that. Now, in prokaryotes, there is an additional complication that we do not see in eukaryotes, because in prokaryotes, the DNA is sitting in the cytoplasm. And as the DNA is being transcribed, you can have more than one RNA polymerase transcribing that gene at the same time. And that's what we're actually seeing here, a gene that's being transcribed by 1, 2, 3, 4, 5, 6, 7, 8 different RNA polymerases, so that we're making 8 different messenger RNA molecules at one time. Do you get that? What's really confusing about that is in prokaryotes, this messenger RNA that's still being transcribed, that is attached to the DNA polymerase, the RNA polymerase is still attached to the DNA. So the messenger RNA is still being made. In bacteria, the ribosomes can bind to the messenger RNA as the messenger RNA is still being transcribed. Everybody see that? In bacteria, while the messenger RNA is being transcribed, ribosomes can bind to the messenger RNA and begin translating it before transcription is finished. Why this happens is in bacteria, the DNA is in the cytoplasm, so transcription is happening in the cytoplasm, and that's where translation can happen. So the ribosome just binds to the messenger RNA and begins translating it before transcription is finished. In eukaryotes, you'll never see that because transcription begins and starts in the nucleus, and then translation happens in the cytoplasm. Any question about any of that complication? All right. So that's a complication that's more complicated in bacteria than in eukaryotes. But eukaryotes is pretty complicated too, and that is you can get more than one messenger RNA being made from the same gene at the same time. And then when those messenger RNAs go into the cytoplasm, you can have more than one ribosome bind to that messenger RNA. All right, any questions about any of that? The next slide we've already talked about. We mentioned that in eukaryotes, you have a processing of that initial RNA transcript. And you have to remove the introns from the transcript of RNA in a eukaryotic cell, and that we call splicing. And then the spliced final messenger RNA can leave the nucleus. the earlier RNA transcript that has the introns cannot leave the nucleus. And this is only seen in eukaryotes because they have a nuclear membrane. And eukaryotes also have introns. Bacteria does not have introns in a gene. All right, any questions about RNA transcription or RNA translation into protein? If not, let's take a look at the little summary. We talked about the structure and function of the genetic material, talked about the flow of genetic information, and it can happen in 3 different ways. We talked about DNA replication, RNA transcription, and protein translation. Now let's move on to how genes can regulate the expression of a gene. We're going to look at that with bacterial gene expression, although it's similar in eukaryotic gene expression, and that involves an operon. Later, we'll talk about mutations, genetic transfer and recombination, and genes and evolution, and we'll probably not get to that today. Well, we might get to it in the lab, not in the lecture. So let's talk about regulation of bacterial gene expression. Cells want to regulate their contents, and an easy way to regulate their contents is to control gene expression. There are some genes, like a constitutive enzyme, that are expressed at a fixed rate all the time. And these we all call constitutive genes, or genes that are always turned on. There are other enzymes that are expressed only as needed. And they can be turned on or they could be turned off. And these are the repressible and the inducible genes, such as a repressible and an inducible enzyme. What's happening is the cell, when it doesn't need the protein, will turn the gene off. And when it needs the protein, it'll turn the gene on. Any question about that? You can regulate a cell in other ways. You can regulate by pre-transcriptional control. Actually, that's what we're going to talk about, pre-transcriptional control, where the inducible operon controls the gene. We can also control that by a repressible operon, but we're not going to talk about that, and you're not going to be tested on repressible operons. You can control a gene by epigenetic control, and these, I don't think your book talks much about them, and you're not going to be tested on that. And obviously, we're not going to cover it. It's actually kind of hairy, epigenetic control. And then there's post-transcriptional control mechanisms where the gene is made, but it's turned off. The protein is made, but it's turned off, and then the protein doesn't function in the cell. So that we call post-transcriptional control. We're not going to talk about that. You're not going to be tested on that. Any question about any of that? So there are different ways that the gene can regulate its contents. And we're going to talk only about an inducible gene. So gene control, meaning the expression of a gene, can be controlled by an operon. We're going to talk about a bacterial operon, which is similar to a eukaryotic operon. It does contain the gene of interest, and that gene of interest is called a structural gene. All bacterial operons have at least one structural gene, Some bacterial operons have more than one structural gene. Usually when we're talking about a gene, we are talking about a structural gene. Like when we talk about the blue-eyed gene or the brown-eyed human gene, that is a structural gene. When we're talking about the gene that codes for the enzyme lactase, that is a structural gene. We're going to be talking about the lac operon, which does encode the lactase gene, and it is the operon responsible for metabolizing the sugar lactose. And when we look at the lactose operon, it does encode 3 structural genes, Z, Y, and A. Z is the gene that we'll talk about the most, and it is the gene encoding the enzyme lactase. But the lac operon has 3 structural genes on it, the enzyme for lactase. It also has the Y structural gene, and the Y structural gene encodes for the protein, transport protein that is on the cell membrane that allows the sugar lactose to enter the cell membrane. And then the lac operon includes the A structural gene, and the A structural gene is a gene involved in converting the sugar beta galactose to the sugar glucose. So the enzyme lactase will degrade the sugar lactose into beta galactose and glucose. Glucose the cell can use. Beta galactose has to be converted to the sugar glucose. And the A gene helps in the conversion of beta galactose to glucose. Okay, any question about the structural genes of the lac operon? The structural gene, or the operon, I guess I should say, contains more than just the structural genes. It also contains the DNA controlling regions that control the expression of the structural genes. And these controlling regions are mainly the promoter and the operator. There is one other region called the terminator, and we've actually talked about the terminator. That's what tells the RNA polymerase to fall off the DNA. And that is part of the operon too. So the operon has the structural genes of interest, as well as the controlling regions of DNA that control the expression of the structural genes. Any question about any of that? All right. Now, in a eukaryote operon, they're similar to this, except the eukaryote operons only have one structural gene in the operon. Everyone got that? So in a sense, they're simpler because there's only one structural gene on each operon. However, eukaryotes have more control of the structural gene. There are more regulatory regions and more proteins that control the expression of that operon. So in that sense, it's more complex. Now, we digest the sugar lactose, assuming you can. And so in eukaryotes, we do have the Z, the lactase enzyme, the Y, the permease enzyme, which is the transport protein that brings the sugar lactose into the cell. And then the A gene, we all have that in eukaryotic cells. However, we have it in 3 operons for eukaryotes, not in 1 operon. Everyone got that? Because in eukaryotes, there's only 1 structural gene on each operon. All right. Let's talk about how the LAC operon works. And I'm probably going to run out of time because I'm going to go over. The LAC operon is an inducible operon. That means the gene is normally turned off, but it can be induced to be turned on. What normally happens is there is another gene which is not part of the LAC operon. And let me go back here. It's this I region. It is not part of the LAC operon. It has its own operon. And it encodes a repressor protein. And this repressor protein is active and will bind to the operator. Now, when the RNA polymerase binds to the promoter at the LAC operon, it starts moving down the DNA and then stops at the repressor protein. So that's what this gene, I gene, does. It encodes a protein which regulates the LAC operon, and this protein binds to the operator and then starts RNA polymerase so that there's no transcription of the structural genes in the LAC operon. Okay, any question about any of that? Normally, the regulatory gene is not found near the gene it regulates. But in the case of the LAC operon, the regulatory gene is right next door to the LAC operon. In eukaryotes, the regulatory gene is usually on another chromosome than the gene that it regulates. All right, any questions about any of that? So how does the gene get turned on? Well, we have an inducer, which is actually allolactose. Allolactose is an isomer of the sugar lactose. Whenever lactose is around, you'll have allolactose around. They're just, they're the same molecule, they just have a right-handedness and left-handedness. Whenever allolactose is around, it will bind to the repressor protein and inactivate it. When the repressor protein is inactivated, it cannot bind to the operator. If it doesn't bind to the operator, then when the RNA polymerase binds to the promoter, the RNA polymerase will run down the DNA, go past the operator, and then transcribe the structural genes in the LAC operon. Okay? And you'll notice in the case of the LAC operon that we have an unusual messenger RNA that we've never seen before. And this messenger RNA codes for 3 proteins. Beta galactosidase is another term for the enzyme lactase. Permease is the protein, the transport protein that brings the sugar lactose into the cell, meaning it's the transport protein that allows lactose to come across the cell membrane. And then transacetylase converts beta galactose into the sugar glucose. Okay, any question about any of that? So allolactose, whenever lactose is around, will inactivate the repressor protein, and then that will turn on the gene. However, we have a question here. And that is what happens to the repressor protein that's already bound to the operator. How does that come off? Anybody have a guess?
Does lactose bind to it?
That's a good guess, but no. When the repressor protein is bound to the operator, it's in a conformation that the allolactose cannot bind to it. Okay. What happens is that this binding is not permanent. It's binding in a stoichiomic reaction, and every once in a while it falls off. If there's no allolactose around, either it or another repressor protein will bind to the operator and then shut down this gene. However, if allolactose is around and the protein falls off, allolactose will bind to the repressor protein and inactivate it. And so if ever allolactose is around, it will turn on the lac operon because the repressor protein will eventually fall off, and then it will be bound and inactivated. And then the RNA polymerase can bind to the promoter and then run down the DNA and transcribe the structural genes. Okay? Any questions about the lac operon? No questions? Well, here's a question for you. This RNA polymerase, what happens to it? Is it stuck here forever as long as the repressor protein is bound to the operator? What do you think? Is that RNA polymerase stuck there forever?
No,
Come
it's
on.
not stuck there forever.
Say again?
No, it's not stuck there forever.
Yeah, that's the correct answer. The RNA polymerase binds with the stoichiomic reaction, and every once in a while, it'll fall off the DNA, releasing it from this conformation where it's stuck with the operator. And then it'll bind on another promoter and try and transcribe that promoter, that gene. Okay, so that's the way the lac operon operates. Why do you think the lac operon is an inducible gene where normally the gene is turned off? What do you know about the sugar lactose? In E. coli, how often is E. coli going to see the sugar lactose? Where do you see the sugar lactose? Come on, you guys know this. Where do you find the sugar lactose?
Fermentation?
Well, yeah, but I'm talking about the sugar lactose. What food is it in? Where do you find it on Earth?
Oh, milk.
Yeah, you find it in dairy products and milk. How often is E. coli going to be around dairy products? It's either often or not very often. So what's your guess?
Often.
Why do you say that? All right, not getting anyone to reply. If E. coli is on the ground or on the counter or on an apple on a tree, it's not going to see the sugar lactose. The only time E. coli is going to see the sugar lactose if it's in a dairy farm or in somebody's gut who drinks and eats dairy products. Okay. All other times, E. coli is not going to be seeing the sugar lactose because the sugar lactose is not a very common sugar. It's only found really in dairy products. And so that's why E. coli is an inducible gene. It shuts down this gene because most of the time, E. coli is not going to be seeing the sugar lactose. And you want to shut down the gene if you're not going to be seeing the sugar lactose, because that would be energy efficient. If you make the lactase enzyme and all the other structural genes, the cell is going to be wasting energy. And so that's why this is an inducible system, because E. coli is not usually going to be seeing the sugar lactose. But if it sees the sugar lactose, it can be induced by allolactose to turn on the gene. All right, any questions about the lac operon? If not, I will see you at 6:30 for the lab.